Well, this is the end of our business meeting and the beginning of a very important session, which is George Palade lecture, and I will ask the president of IAP, Dr. Suresh Chari, to come and chair this session. Please, Suresh.
Thomas here as well. Good afternoon, everybody. It's on behalf of my co-chair, Professor Thomas Grass, who is the incoming president of the International Association of Pancreatology, and myself. It's our honor and privilege to invite Dr. Marcus Lurge to deliver the George Palade lecture. You heard Dr. Saluja's felicitation of Dr. Marcus Lurge yesterday. I will briefly go over his background and that of Dr. Palade in case you were not there yesterday. Professor Palade was a Nobel Prize winning cell biologist who made significant advances in understanding of the protein trafficking in cells, acinar cells, which has led to a legacy of pancreatologists interested in cell biology, including Jim Jamieson, Fred Gorelick, and all of them are from that lineage, and has had a significant impact on pancreatology. And in his honor, the International Association of Pancreatology started an award for excellence in science. And though this is not given every year, this year is our honor and privilege to award this to Dr. Marcus Lurge. He himself was born and brought up in Germany, went to medical school in Germany, and then joined Ashok Saluja at Harvard for doing his first studies on the pancreas. We heard about the opossum stories and how he did pancreatitis work on opossums there. Then moved on to Italy to get some experience in electron microscopy. Went to Germany to train medicine and gastroenterology at Ulm. Did his initial faculty position at Münster, and finally is professor and chair of medicine and gastroenterology at Greifswald University. And his contributions to pancreatology are legendary. They are reflected in the number of publications he has, the mind-boggling numbers that Ashok mentioned yesterday, including 500 plus publications and 30,000 citations, and all of them are like in figures that are mind-boggling. So let's just say an immense contribution to the literature. But what impressed me, has always impressed me, has been the ease with which he gives both a clinical talk and a basic science talk. It's a pleasure to listen to both of them because he makes them both understandable to people in the other universe. And that's because he himself lives in two universes and with ease. He does the RCPs on one hand and he does fairly basic work on the other, and is completely comfortable in universes which are quite separate and different. I had the pleasure of reviewing one of his most recent papers in gastroenterology that showed that even pancreatic dysfunction, not insufficiency, is a significant contributor to the change in the microbiome in subjects who have deficient pancreatic function. So he continues to do some really cutting-edge work that is going to impact how the field moves forward. So for us in the IAP, as I said yesterday, it is our honor to honor Dr. Marcus Lurge for this award. So on behalf of IAP and my colleague, Professor Thomas Grass, I'd like to invite you over for this one, if you want to say a few words here.
All right. Actually, Suresh, you said everything that I could have said, besides the fact that we worked together 2 years in Ulm, and since then we've been friends. and you've been a mentor for me, and thanks for that. And I'm really looking forward to your talk. I've seen it evolving over the last 3 days. I was sitting beside you, so I'm really curious how it looks like.
Thank you so much, Suresh and Thomas. Ladies and gentlemen, it's a great honor to be here today for this lecture and this award. I have to tell you though that what Ashok told about me yesterday, he made up essentially while the slides prepared by someone else were given. It was greatly exaggerated. Even the numbers are greatly exaggerated. My goal in this presentation is to convince you that unlike the impression Ashok gave, I'm not retiring anytime soon, at least not for the next 5 years as my lab is well funded. I have no real disclosures to make in the connection of this presentation. This is the first paper I was given by my mentor Peter Hopper-Seiler in Freiburg when I started my medical thesis. I had a background as an art history student before I went into medicine, and this highly geographical arrangement here of the pancreatic acinar cell totally fascinated me. I thought it was artwork. And this paper actually is penned by Jim Jamieson and George Palade, and essentially it convinced me to not only go into the pancreas field or stay in the pancreas field and to train with Mike Steer, but also to want to learn electron microscopy, to do things like this. And that's why I did an internship in pathology where I did electron microscopy. And my postdoc, I spent some postdoc time with Jacopo Mildalesi, who is a student of George Palade. And he was there together with Jim Jamieson. And I think you will forgive me if I remind you briefly of Jim Jamieson, who I did know, and Fred already gave a tribute to yesterday, because his papers left such a lasting impression on me. Jim died 10 days ago at the age of 84. He's Canadian. Interesting that the early cell biologist, that's true also for Keith Porter, who invented the microtome. They were both Canadians from different sides of Canada. He did his PhD with George Emil Palade at the Rockefeller and moved with George Palade to Yale. And when George retired, Jim Jamieson took over the chairmanship for many years, and again as interim chair before Jim Rothman could decide to join the Yale department. Most of his seminal work is about what I will also be talking about today, endocytic vesicle membrane retrieval at the pancreatic acinar cell lumen and the role of the Golgi complex. I must say that the first recipient of the George Palade Prize by the IAP was Jim Jamieson, and I feel humbled to stand in the same line and on the same podium here. Essentially, I will focus today when I talk about cellular pathogenesis, and as Suresh said, I will switch between clinical and cell biological work because it all feeds into the cellular pathogenesis about gallstone-induced pancreatitis. And some of the work, the seminal work regarding gallstone pancreatitis is actually quite old. This is a terrific study by an Australian surgeon, Juan Acosta, who took the stool of patients and washed it like a gold digger until he found tiny goldstones. And that's something no other postdoc has ever attempted to repeat this study. It appeared in the New England Journal many years back, and what he found was quite important. Gallstones retrieved from the feces of 90% of patients with biliary pancreatitis, and in only 10% of patients with symptomatic gallstones were found in the absence of pancreatitis. So it's the migrating stone that causes pancreatitis, and it's not a gallstone sitting somewhere in the gallbladder. That was important, and the theories why this migrating stone would cause pancreatitis are threefold and very confusing. Confusing because this man, Eugene Lindsay Opie, a pathologist in New York, he published in the same year, in the same journal, two hypotheses why gallstones cause pancreatitis, and they're mutually exclusive. Number one, that's his own drawing, I think it's better than mine, is that what the gallstone does is obstruct the pancreatic duct, and that's the cause of pancreatitis. This second hypothesis from another patient, and he followed these up in animal experiments, is that the gallstone impacts at the papilla, creates what he called a common channel behind it, through which bile would flow into the pancreas and trigger pancreatitis. And that's a hypothesis that's very attractive to surgeons who, until 20 years ago, operated on patients with gallstone pancreatitis and found a bile-stained pancreas very late into the disease course. For a number of reasons, this is an unlikely hypothesis, and most important is the work of Jean DiMagno, who said that this papilla is usually too short for a gallstone to create such a common channel. A third hypothesis was created in Frank Moody's lab using the opossum, I'll get to that a little later, which said that it's the pancreatic duct's obstruction pathogenesis, but if you have bile occlusion or cholestasis, then it aggravates the disease. Well, this was my task in the Boston lab when we started, and Mike Steer and Ashok Saluja put me to work with opossums, and I can assure you they're very unpleasant animals, particularly if you want to convince them to volunteer for biliary pancreatitis experiments. But they have two distinct advantages. One is if you ligate them at the papilla, they develop a very severe form of pancreatitis, which is indistinguishable from human necrotizing pancreatitis, even in the early phases, with a lot of hemorrhage and a lot of necrosis. So the other advantage is, even if you're not a Japanese or Chinese surgeon with tiny hands and good skills, it's very easy even for a dumb German gastroenterologist to ligate the ductal system. It's very much like the human system, and you, by ligation, you can generate situations that mimic either pancreatic duct obstruction or bile and pancreatic duct obstruction or a reflux situations into the pancreatic duct. And the first theory I had to test was, where does pancreatitis begin? At the time, Günther Kloppel and others had said that pancreatitis begins with hemorrhage, and not in acinar cells, or with lipid necrosis in the peritoneum before the acinar cells. And the answer was quite clear. We did this over the first 24 hours in the opossum, and it was very clearly the acinar cell where pancreatitis was initiated. That was a very important finding at the time. Now no one talks about these other hypotheses, and it's very clear that the acinar cell is where pancreatitis begins. The next thing we tested, and that was probably even more important, was, is it the pancreatic duct obstruction? Is it the reflux? Or is it the combination? And if you look at early necrosis or late necrosis, at early necrosis it's very clear over the first even hours, then days, this is up to 7 days, it doesn't matter which one you ligate. So the conclusion from this experiment was very clear. It's the pancreatic duct obstruction that triggers the disease. At later time points, it seemed a little, this was the only significant difference, that an additional cholangitis or bile duct obstruction aggravates the disease somewhat, and this is what we believe clinically also. Then Michael Runze, also in the lab, well, they only let Germans work on the opossum because no one else would ever touch one. He tried to relieve the pressure by putting a balloon in. And what happens is we were very, very positive about this finding, and we said, if you take the balloon out, there's no further progression. So this was ligation for 1 day, 3 days, 5 days, and if you took the balloon out after 1 day and left it out for 7 days, there was no progression. My conclusion today is something different. There was no reversion of pancreatitis. So whatever you did, relieving the pressure, even after 1 day, did not revert the disease for the next 7 days. And this is the same finding the Dutch had when they just completed the big APEC trial. I'm grateful for Nicolene Schepers who just gave me the slide. This is unpublished data and was just presented at United European Gastroenterology Week. They screened 1,200 patients to find 232 they could include into an ERCP study. And they were very ambitious. They said, we want to do an ERCP with extraction of stones 24 hours within 24 hours after admission, or within 72 hours of symptom onset. That's extremely ambitious, and you have to have 5 times as many patients as you then, because everyone else comes later. It's also important that they excluded everyone with cholangitis, because it's very clear now that with cholangitis, remember in the opossum the additional effect on severity, you have to do an early ERCP. And the results was very disappointing. There's no difference in death, there's no difference in the composite end-pound, and there's no difference in new organ failure. What do we learn from that? Once pancreatic duct obstruction, and this is mostly transient pancreatic duct obstruction, starts, the disease is on its way. And even taking the obstruction out as early as 24 hours after admission will probably not change the natural history of the disease, even though it's very important to take the gallstone out if it causes cholangitis or cholestasis. There was a fourth hypothesis, which feeds into the other two. 2 Australian surgeons had suggested from dog experiments what could happen is that the gallstone passes through the papilla and rendered the papilla insufficient, allowing pancreatic juice with duodenal content to flow into the pancreas. And I had the opportunity to test that together with, an Argentinian surgeon, Carlos Hernandez. This is at the time before ERCP. He had collected 3 groups of patients, all of which had undergone cholecystectomy, and all of them had a T-train put into their bile duct. A T-train put into your bile duct means there cannot be no pressure of biliary pressure or reflux into the bile, and this allowed him to feed these patients a number of days after the T-train put in, and after the meal stimulation, he measured amylase in the T-drain outflow. And there was a very clear difference. Gallbladder stone patients had no reflux of pancreatic juice into the bile. Common duct stone patients had significant flow into the bile, and gallstone pancreatitis patients had the highest reflux, which means that after passage of a gallstone, these are the most likely to have passage of a gallstone. There is sphincter stenosis and not sphincter insufficiency, allowing for reflux of trypsin and duodenal content into the pancreas. That was a very important finding, and that's why it was accepted at the Lancet. It's very difficult to prove that in patients, particularly in the day and age of ERC, where you can easily take endoscopically the stones out. We found some patients that fed into this hypothesis. One is a young girl of 16 years old. She came for complications from her choledochal cyst, but she had perfused her pancreatic duct with bile for at least 16 years, because this is an inborn error. And she never developed pancreatitis, meaning that at regular pressure, bile into the pancreatic duct does not cause pancreatitis. This is another patient. This is a young woman I met in Ulm. After delivery of her baby, she developed cholecystitis. And the surgeon in a small hospital who didn't have ERCP available took the gallbladder out and put a T-drain in, just like the patient in the Argentinian cohort. And with the T-drain in, she developed pancreatitis, meaning that what the stone had done, he had obstructed the papilla, obstructed the pancreatic duct. When we took the stone out, this is from this patient, when we took the stone out, you see pancreatitis subsided quickly, and when we had a meal stimulation, because of the papillotomy, there was no reflux of amylase back into the T-drain. And taken together, the opossum experiments with the human study and the anecdotal cases, the pancreatic duct obstruction pathogenesis of gallstone-induced pancreatitis became textbook knowledge. I was very proud at the time when Peter Banks, who is in the room, put this into his Schlesinger and Fortran chapter, and you can imagine for a young man that's like a knighthood when you get that. Yet, somehow the bile reflux hypothesis survived, even though it's very unlikely, there's no positive evidence for it. Why is that? There's a number of reasons. One is a historical one. This is Claude Bernard, probably the most eminent 19th century physiologist from Paris. And in his Mémoire sur le pancréas et sur le rôle du sucre pancréatique dans le phénomène digestif, he generated biliary pancreatitis by injecting into the pancreatic duct of dogs and of rabbits bile. And what he reported was severe pancreatitis. And you will know, Hannah Algil gave a talk yesterday where he introduced his pig pancreatitis. What do they do to make severe pig pancreatitis? They inject bile into the pancreatic duct. Even though that never happens in human and it's not the cause in human, it's a very safe way of generating severe pancreatitis. Another factor didn't help. That's the discovery of the bile acid surface receptor. 10 years earlier, the farnesoid X receptor, long predicted by Alan Hoffman, was found, which is a bile acid receptor in the nucleus of cell. And 10 years later, this G protein coupled bile acid receptor was found on the surface of cells. So bile acids have a signal transduction function. And then suddenly, very bad for my hypothesis happened. This bile acid receptor in the pancreas sits at the acinar lumen. And the only explanation can be to cause gallstone pancreatitis. So there must be bile reflux if the receptor is there, and that must cause pancreatitis. At least that's the way George Pancreatitis and Mike Steer saw it. They used the bile acid receptor knockout mouse, and when they infused bile acids into the pancreatic duct of these animals, it caused pancreatitis. It caused much less pancreatitis in animals with a knockout of the GP bile receptor. That the same was not true for cerebrally-induced pancreatitis, where a bile acid receptor is not supposed to play a role. And the same was true for isolated pancreatic acini. If you expose them to TLCS, which is a bile acid, they activated trypsin and they underwent injury. Not so in the knockout acini, and as a control they used the cerebrolin. So we found it very hard to argue. This is a very pervasive evidence. So if the receptor is there, the bile acids must find a way of getting there. And we argued that there are other ways of bile acids to damage the acinar cell. In addition to the apical G protein coupled bile acid receptor, there is even an apical sodium-dependent co-transporter for bile acids. But more importantly for us, or our argument, there is also a lateral bicarbonate-dependent bile acid exchanger. So the bile acids could get to the pancreatic acinar cell even from the basolateral side. That means cholestasis from the blood. And they do a lot of damage by different ways, by release of calcium from intracellular stores. This is mostly the work from Ola Peterson's group, by inhibition of the SERCA pumps, activation of PI3 kinase, activation of the ryanodine and the IP3 receptors. So there are different ways of bile acids without using or having the need to get to the apical lumen of doing damage in the acinar cells. Back to the opossum. We put a lot of work into finding why does the pressure in the pancreatic duct, without the kneel of bile, induce pancreatitis. And one experiment again in the opossum, we tied the bile duct off, so there can't be any reflux, and then we perfused at low pressure markers of endocytosis into the pancreatic duct. And we used markers for fluid phase endocytosis, which is either radioactive dextrane or simply 40 dextrane, 40,000 molecular weight, or we used markers for receptor-mediated and charge-coupled endocytosis, which is horseradish peroxidase or cationic ferritin. You saw an elegant talk by Olga yesterday where they used this on the fluorescence microscope level. We couldn't do that at the time. That's why we used the electron microscope. And this is the only time I used Jim Jameson's technique of autoradiography electron microscopy. I do not recommend doing that. It is extremely messy and extremely cumbersome. Nowadays, you use fluorescent markers. But what was interesting, we didn't see much change in the charge-coupled and in the receptor-coupled endocytosis. But in the fluid phase endocytosis, which is something equivalent to the trypsin secreted into the lumen, it's taken up again, And it's under physiological conditions, the DEX strain, either radioactive or C14 labeled, or are visible on EM, like here, it appears in the secretory pathway. So what's taken up by the cell is recycled by the cell from the acinar cell lumen and brought and used again and goes into the secretory pathway. Not if you obstruct the duct, this is 1 hour and 3 hour, then it goes to what we call the lysosomal, what you would probably call today the lysosomal autophagosomal pathway. So the conclusion from this experiment, in the absence of bile and with just pressure, you get a profound missorting of endocytosed fluid phase markers from the secretory compartment to the lysosomal autophagosomal compartment. So that was a clear indication that pancreatic duct pressure is the cause. Returning to Germany, I abandoned opossums forever and never seen one other than as roadkill in the United States. And we went to work with rats and mice. And again, we used pancreatic duct obstruction models. The one we used then was a simple pancreatic duct obstruction where the complete pancreas is tied off for very brief periods of time and without excessive pressure. And what you see is that you do get something that looks like pancreatitis with high amylase, but also with high trypsin activation. So where does that come from? That pancreatic duct obstruction causes trypsin activation, and what we looked into is the calcium signaling in pancreatic duct obstructed animals. And we obstructed the duct for a very brief period of time, then we took the acini out and we measured calcium in these acini. Usually the calcium signal is like a peak, and then you have oscillations and a plateau. And what we found is that in these animals, you get increasing intracellular calcium concentrations depending on the duration of the pancreatic duct obstruction. Again, no bile acids near them. And when you looked at that and quantitated it, it was very clear that the regular physiological oscillations came down and the pathological signals went up. And that told us there must be a signal that is triggered by pancreatic duct obstruction, and that leads to a default calcium signaling, and we didn't know what it was. And the discovery was made just recently, and this is the answer to it. This is piezo, a receptor discovered about 8 years ago or 6 years ago. And piezo is a very interesting receptor. It's pressure sensitive, it's mechanically activated, and it looks like, if you're French or from Brittany, it looks like one of the oldest Celtic symbols, the treskel, that when you push onto any of these blades, then it gives a signal, and it's a mechanically activated, unspecific cation channel, but it has a preference for calcium, by the way. And its only two ligands are called Yeti and Yoda. I assume that the postdoc in the lab looked a lot of Star Wars before naming these ligands. And the lab to find out that this is relevant to gallstone pancreatitis is Joël Romac in Roger Little's lab, who is here in the room, and I'm proud to present some of your data to it. What they did is they didn't ligate the pancreatic duct of mice, but they put a very sophisticated pressure probe into the pancreatic duct, which allowed them with very low pressure to infuse saline into the pancreatic duct. And they did this at an increased pressure. This is the usual pressure is 11 millimeter mercury, and they did it as an increased pressure, and what they got is very similar to our observation: high amylase, high myeloperoxidase, that means inflammation, and high damage on the histology score. And then they used an antagonist for the piezo receptor, and that is GSMTX4. It's from the tarantula spider. That's one of their poisons, the only, to my knowledge, known antagonist of the piezo receptor. And what they found is adding this antagonist reduced the amylase, reduced the MPO, and reduced histology. That was already sufficient evidence for a role of the piezo receptor. Then they generated an SNI-specific knockout mouse for the piezoreceptor. That is necessary because if you're in embryonic development, as soon as you get a cardiovascular circulation and don't have piezoreceptors to regulate pressure, you're dead. So you can't breed global knockout mice without piezoreceptors. They made a piezo SNR cell knockout, and you see again, not so much on the amylase, but on the MPO level, on the histology, the pressure-induced pancreatitis was greatly reduced. Then they used Yoda, the agonist, and Yoda alone could increase, could kind of mimic the pancreatitis phenomenon, but not in the knockout mice. And this is most important to me. You have a pathological calcium signal induced by Yoda and inhibited by the tarantula inhibitor GSMTX4 of the piezoreceptor. So you get a pathological calcium rises, and you can inhibit them with the inhibitor. That may explain what we never found, the calcium signaling disturbance that is caused by pancreatic ductal pressure. So my interim conclusion, before I get to the rest, is that gallstones cause pancreatitis by transient pancreatic duct obstruction. It's mostly transient, but it's sufficient to trigger the disease. And it's mediated by pressure-activated piezo1 receptors, that is mechanically activated cation channels with a preference for calcium. It triggers a rise in intracellular calcium concentration and trypsin activation, and followed by cellular tissue injury. But how does the calcium induce the trypsin activation? And that was a chase we were involved in for a number of years, but before we could answer that question, we had to develop a number of technologies. And I'm proud to say that we are the first to do so. This is an SNR cell, slightly flattened under the microscope, and what you see is this is not immunostaining of trypsin. This is actually a fluorochrome, an isoleucine proline arginine substrate, which when activated shows you where the active trypsin sits. And Burkhard Krüger, one of my close collaborators over the years, developed this technique not only for the microscope, but also, of course, to do in fluorometers, so you can quantitate the trypsin activation in SNR cell, but you can also localize it. And you can pinpoint exactly where it sits in cells. However, light microscopy is still somewhat crude if you don't have a dual photon, which we didn't have at the time, so we developed a second technique. This is the trypsin the trypsinogen activation peptide, to which a company in England has once developed an antibody, and which we used for immunolabeling for electron microscopies. These are cryosections of mouse pancreatic SNI. And what you see here, the little gold dots, this is the technique I learned in Milan, show you where the active trypsin sits inside the SNR cell. Another technique that came in handy to localize the signal. And one question, or the first question, was, where does the activation begin of trypsin? And this is something I borrowed from Mark Sherwood's a paper in PNAS, and he added this little video, and it does work, thank you very much. And what you see here is an SNS fed with rhodamine. Can you click on it again? Okay, it doesn't matter. I can show it here. So over the time course, what Mark Sherwood showed is that you have large endocytic vesicles from the SNR cell lumen. That's what we try to show with the electron microscope. And then eventually, this is a compartment in which you can demonstrate active trypsin using the same technology I've shown you before. And to us, that was a very late phenomenon, because the Ole Pedersen group could only show this autophagosome-associated trypsin activation very late in the process. And so we tried to look at much earlier time points, and we used knockout mice which are positive for LC3. LC3 is an autophagosomal membrane marker. And it only lights up in the cell when the cell forms autophagosomes. And it doesn't light up as long as the cell doesn't have autophagosomes. And we co-localized this with the trypsin signal, and to make a long story short, we could find ample evidence in single-cell measurements where you had autophagosome formation very early, but no trypsin activation. But this is more important, you found plenty example where you had trypsin activation without autophagosome formation, or this is more classical, you first have trypsin activation and then you have autophagosome formation. We try to confirm this by electron microscopy, and again using the TAP technique. Very early, the one before was SNI. Now this is in an animal stimulated with CCK. You see these large gold granules here. That's the lamp, the marker of autophagosomes, in beautiful autophagosomes which are characteristic in their double membrane. And you find in this picture exclusively the trypsin activation peptide in what's looking like a secretory vesicle. I may point out at this point that this paper is just a JBC in revision. I know the reviewers in the room. Have mercy and accept it. I think the revision is okay. So how does this get together with the calcium signaling? And maybe we can click on these. Yeah, not quite. What this was meant to show you, the first one is the calcium signal inside the acinar cell, which is over minutes, and this which is over seconds, and this is over minutes. We use this co-labeling to go to individual acinar cells and look for each of them, which ones activates trypsin and which one doesn't, and what the corresponding calcium signal is in this one and in this one. And what we found was that the trypsin activation is always associated not with classical oscillations in calcium, but with a sharp rise and a subsequent very high plateau. We later found that the sharp rise is not so important, the extent of the rise, but the sustained calcium plateau is. And I'm proud to say we were the first to publish this signal that leads to trypsin activation. It was independently found, of course, in Ole Peterson's lab. We were just very lucky that our paper came out 3 months earlier than theirs, but Ole is gracious enough to acknowledge that. It doesn't happen very often that you're on calcium signaling a little earlier than Ole Peterson has. The field has moved, so this is not the only important signal. What we knew at the time, what we didn't know at the time, was the importance of store-operated calcium channels. We did know about the IP3 and the NADP, the ryanodine receptor and the IP3 receptors, and there are other mechanisms that regulate the calcium inside the cell. What is probably not important in the pancreas is the sodium-calcium exchanger, which is not expressed in acinar cell but in other cells. But what is very important, plasma membrane calcium-activated ATPase, that is this mechanism, and tomorrow you'll hear a talk from Joseph Mallet's group in Szeged, where they found how this leads to a triggering of pancreatitis. How do we antagonize this? So you can't give a person a calcium chelator. We did that in animals, but they're so highly, and you could abolish pancreatitis by a calcium chelator like Bapta, but they're so toxic that they would never be used in humans. So one of the persons in our lab, Frank Morin, was very interested in magnesium. He just came from a Max Planck Institute in Dortmund. And so we tried to co-localize the calcium and the magnesium signal in acinar cells. And what we found is that at the apical pole in the rise, we used Fura2 as the signal for calcium and MagFura2 as the signal for magnesium at the time. Essentially, to bring this to a conclusion, calcium rise is initiated at the apical pole, and it progresses to the basolateral pole. These are the two poles and the two curves. And the magnesium drop is just the opposite. It's initiated as the basolateral pole and progresses to the apical pole. So what we thought is that calcium and magnesium are exchanged for each other. What if you replace the calcium with magnesium in the acinar cell? This is what happens. This is isolated acini, and with higher concentrations of magnesium you add, you get an abrogated calcium signal. And our thought was, maybe we can use that as a physiological calcium antagonist also in pancreatitis, and we did. This just came out in Gut in 2014. We used 2 different models of pancreatitis where we had fed mice with CHO that was either depleted in magnesium, this is the -300, or very rich in magnesium. And they eat this. The worst you can get from too much magnesium is a little soft stool or diarrhea. So there are no real side effects. And what we found that in the magnesium deficient group, even physiological CCK concentrations induce pancreatitis in the mouse, whereas higher magnesium concentrations abolish even the high CCK-induced pancreatitis. So there was a very clear correlation between the magnesium in your food, and we know that pancreatitis patients have low magnesium levels, and we could abolish here a good example in cerulean pancreatitis. These are the vacuoles you get with low magnesium, and these are the disappearing vacuoles with high magnesium. So this gave us reason enough to believe that this is clinically applicated, and I can say that we have conducted 2 trials on this. One is the EuroPac-2 trial, where we have randomized patients into 3 groups, one with antioxidants, that's a different story, one with magnesium, and one with placebo. We have recruited 288 patients with idiopathic recurrent pancreatitis, at least 2 episodes over the last 2 years. The study will be analyzed. Last patient was in last April, and next year in April we will know the results because we follow these patients for a full year. A second study is underway. We're still in the recruitment phase where we give patients an infusion of magnesium before ERCP, and we'll have to see whether that has a preventive effect. For me, that's a big experience because this is the first time that in my career it was possible to find some phenomenon in the laboratory and bring it into a clinical trial. No matter how this clinical trial ends, this is something that doesn't happen too often in a physician-scientist's career. The last phase of my talk I want to get to is this. Matthias Sendler in the lab, he was not pleased with having mouse pancreatic duct ligation giving only a mild form of pancreatitis. So he wanted a more severe form where, number 1, he can see necrosis, and number 2, he can study the progression to chronic pancreatitis. And he invented this model where we only ligated part of the pancreatic duct and then stimulated with cerulean, just once or twice. And what happens is you don't have biliary impairment. You have a side of the pancreas which undergoes severe pancreatitis and another side of the pancreas which stays completely normal. So you have your intra-animal control, and this is the model we're currently using all the time. If anyone is interested, he can come and learn it. It's very easy to do, and I think it's probably the future of this model. And you can see here that after 3 days, there is a much more severe form of necrosis and pancreatitis than you see in the cerulean model. So why am I telling you this? What he found was that he found this is an area of the pancreas where all the green is intact pancreatic acini because the trypsin is labeled. And in this area where necrotic cells are, you see massive infiltration by CD68 positive macrophages. When he took a closer look, he found that inside these macrophages, he found active trypsin. And sorry, this is not initially active trypsin, this is just trypsin immunofluorescence. And what's the trypsin doing there? And he was very persistent in continuing with this, and he used isolated macrophages, and he exposed them to either trypsinogen or to total acinar cells. And what he found is that these macrophages activate trypsin inside their own compartments. So they take up trypsinogen, or they take up acinar cells, then they activate trypsin, and they do so in a cathepsin B-dependent manner. I'm not going to go into the cathepsin B theory because Miklos criticized it too much yesterday, but it's a very cathepsin B-dependent process, and it doesn't matter whether you give them total acinar cells or if you take isolated cymogen granules. They will take up isolated cymogen granules with trypsinogen. They will then activate it to active trypsin. And do they just digest it? No. The answer is they themselves become activated. Here is the CD68 plus cells, and you can see this. You can inhibit it with nafamostat. You can inhibit it when you inhibit the uptake with bafalomycin. The same happens. So to make a long story short, upon trypsin activation, macrophages undergo activation, and they produce cytokines like IL-6, MCP-1, and TNF-alpha. And TNF-alpha was shown by Matze again to be, again, an activator of trypsin activation in acinar cells. So the whole thing feeds on itself, and even outside acinar cells, we have trypsinogen activation to active trypsin. I come to my conclusions. Gallstone pancreatitis is caused by pancreatic duct obstruction. I hope I have ultimately convinced at least this room it's not bile reflux that causes this. It's mediated by pressure-activated piezo1 receptors, that is, mechanically activated cation channels with a preference for calcium. That triggers a rise in intracellular calcium concentration and trypsin activation, as shown by Roger Little groups. It's followed by cell and tissue injury, which can potentially be prevented or reversed by replacing intracellular calcium magnesium, and there are 2 trials underway. The most recent development where people try to inhibit the calcium cascade is CRUC channels, the store-operated calcium channels, and they are drugs that are currently being in clinical trials to test to abolish the pathological calcium signal in a different way than we are trying here, but with a more advanced technology. And finally, in which acinar cell injury constitutes a danger signal specifically for macrophages, which then undergo activation upon trypsinogen activation and thus represent an alternative treatment target to acinar cells. I rest my case. This is Greifswald from the beach. Someone postdoc a palm tree in there, but we had a very hot summer. It's about like November in Miami. And these are the people I have to thank who worked with me over the years on this topic. One is Matthias Sendler, Ali Akdassi, Uli Weiss, and Peter Siemann. Specifically, I want to thank Julia Meierle for having tolerated me for over 18 years before she moved to Munich 2 years ago. I'm happy that today Julia Meierle and Fabian Frost, they're both in the room to celebrate with me tonight the Palade medal. I want to remember Walter Hallang, my collaborator for 20 years on the trypsin question, who passed away on August 20th this year after a 2-year illness which started as necrotizing pancreatitis. He thought it cynical that the disease he researched would eventually kill him, and these are many others: Ashok Shalucha, Miklos, Thomas Wartman, Anna Gukowska, Dave Whitcomb, Mike Stier, Fred Gorlig, and Peter Hedge, who I had the pleasure and fun to work with for many years. I hope I convinced you that I'm going to stay in business. Thank you very much.
Thank you, Marcus, for this amazing story that you have taken all the way from the bedside to the clinic, to the bench and back again to the clinic. As a award lecturer, there are no questions here for you. If you have any for Marcus, please meet him afterwards. But I would invite Pramod, to our Secretary of the International Association of Pancreatology, to hand over the award to Marcus.